Okay folks, so this is a quick review about the central dogma. And the whole point of the central dogma is how can we go from DNA to protein. So DNA have all the information to decide about one organism. So anywhere between being a normal person or a dog to a weird creature like this, or a weird fish that look kind of like this. Well, DNA decide all of that. Now the question is, how do you go from DNA to a certain phenotype like we've seen? So really quickly, you pretty much just going from DNA to RNA, or more specifically mRNA, to protein. Alright? And there is a key thing that you need to know is the sequence. So essentially, the sequence means the order at which each nucleotide is going, or the order at which amino acid is going, will make the difference in what you see. So let's give an example. A DNA would have a sequence on order of A, T, G, 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 C, G, A, for example. So this is a DNA. Now, DNA have two strands, as we learn. So I'm going to draw two strands. That means that if one DNA have this sequence, this order of nucleotide like so, the other strand of DNA has to have a complementary to that. So A complementing to T, T complementing to A, G complement to C, G, C, 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 okay, and then C complement to G, like so. Okay, now since there are two strands of DNA, and only one of them is going to be used to make RNA, what we're going to say is that one of the strands is going to be called a sense strand, and the other one is going to be called antisense. Alright? So that means that whenever you make RNA, you are going to make something that looks very similar to the sense and something that is complementary to the antisense. So I'm going to make RNA. RNA is only single strand. So if I want to complementary to a T, sorry, let's start over here. Complementary to a T, I'm going to stick an A on it. Complementing to a it's going to be a T. But remember, RNA, there's no T. In place of T, we would use a U. So that would be A, U, G, complementing to C, G, 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 complementing to G is a C, G, and A. Okay, so now we have an RNA with a U here. Alright? And notice the RNA is the exact copy of the sense, which is the T being changed to a U. That's it. Okay? Now, when this RNA, or to be specific, the mRNA, is being sent to the ribosome to make protein, that is when we're going to just use the three bases of each, and that we call a codon, to read this and to translate this into protein. So each of these three bases is called a codon. Three bases is equal a codon. Alright? And then you would use a table to figure out. So you would use a table or circle Usually you're going to be using a table, I do believe. Something that looking kind of like this. And you would use this table to figure out what is your amino acid going to look like. So we had AUG, right? So the first one is A, second one is U, third one is, so you're going to look down that, look in this area, and then here's G. So look over. Okay, looks like AUG is going to give you methionine. Okay, so great. That means that this is a methionine. And then GGG, let's look in the table. So GGG is going to be glycine, and CGA is going to be an arginine. So now you have the sequence of protein. Methionine going first, meth, and then gly, and then arg. So now you know how the protein is going to be ordered, basing on your original message inside the DNA. Alright? Now let's see, if you were given a question where they give you the sense strand of DNA and they're asking for the protein, do you have to do all of this? No. If they give you the sense strands of DNA, essentially the mRNA can be similar, so you can just do a shortcut and not look at any of this stuff and just go straight to the protein. If they give you the antisense, then you have to translate that into mRNA and then to the protein. Now the thing that really screw a lot of students up is if the question asks for tRNA. Well, tRNA is a complementary to the mRNA. Hang on. So this is why so if the question asks for tRNA, I'm going to make this a little simple here. If the question asks you for tRNA instead of a protein, then you have to do a little bit more thinking. Well, so tRNA in the color blue here is essentially just something that sit on the mRNA in order to do a translation. So you don't actually have to know the structure of tRNA, but tRNA will have an amino acid on top here and then anticodon in the bottom. So the tRNA will have anticodon. tRNA will have anticodon instead of the codon that the mRNA have. So that means that instead of A, they would have a complementary of it, which is a T. Well, but there's no T in RNA, so that would be, there's no T, so that would be a U, and then U would be complementing to an A, and then G complement to a C. So if they give you this and they ask you, in short, they ask you for the tRNA, what you would end up doing is just find the mRNA and then make a complementary. So A would be U would be matching with A, and then C, G, C, U. This would be the tRNA. Alright? And if you notice, the tRNA is exactly the same as the antisense of the DNA. So in short, if you ever get really confused and don't know what to do, just remember this. The sense is the same as the codon. The antisense is the same as the anticodon. DNA has both sense and antisense. RNA only have mRNA only have the sense or the codon. Protein doesn't have codon or sense at all. Protein has amino acid. And the tRNA have anticodon. So tRNA have antisense or anticodon. So then the question is, what's the difference between sense and codon and antisense, anticodon? Why don't we just say sense or sense or antisense? Why do we bother to figure with codon? Well, the technicality is that the codon is only 3 bases, and sense or antisense could be a long string. So when you have this long string of thing, that is called sense or antisense. When you have just 3 bases like I hate this program. When you have just AUG, that's a codon. Okay?